Reactions occurring in plants. In plants, various reactions occur at various phases and various stages. So whenever a reaction will occur, the reaction has to take place with the help of a catalyst. So in plants, we call these catalysts as biocatalysts because they are present in the living system. So usually, a catalyst is something which will convert or enhance the speed of a reaction, the rate of a reaction, so that if something has to take place in say 5 hours, will take place only in 2 hours. So this is because of a catalyst. And when we are talking about enzymes, enzymes are the biocatalysts. So what happened? Biocatalysts occur in living cells and they are able to fasten or speed up a reaction without they themselves having any change. So, in a cell, you have so many components. You know the compositions of the cell. Right, in the cell we have, like if you take a plant cell, we have the chloroplast, we have the mitochondria, where major reactions occurs. That is, in the chloroplast, the photosynthesis occurs. In the mitochondria, the respiration occurs. So when all these reactions are occurring, there will be different compounds which are changed into different other compounds. So this will only take place because of the enzymes. So actually, this study of these enzymes started very long ago. When Louis Pasteur first spoke about the fermenting factor in the year 1860. So for this fermenting factor, Kohane proposed the name enzyme. So which he did it in the year 1887. So for the same thing, there were different works. Like in these days, you have got a net. We are able to talk on the net, and everything is taking place where everything can go from one place to other. To so many media, you have all the media to go. But in those days, a communication from one country to another country is to take a lot of time. You should only write letters. So, or we should go through ships, and when all the other all the other travel mechanisms were not there, which we are having now. So when Edward Buckner was working, he called it as zymase, and the research work continued where J.V. Summer was the first person to crystalline the enzyme. So we will see, when we see about the properties of enzymes, we will know what is crystallization or what is solarization. So all of us know the basic concept that all enzymes are proteins. We know that all enzymes are protein, but all proteins are not enzymes. That's a very common concept which is coming from years together. Proteins, right? So we know that there are different types of proteins. We have proteins in the aleurone layers. We have proteins like albumin we have in the egg, and we have proteins which are enzymes also. These enzymes basically
Excuse me, madam.
Yeah, ma.
Madam, your sharing is. Madam, your slide is only one slide is here.
Okay, 1 minute. You can see the slide now.
Slide. Okay, only one slide we are seeing, madam. Please.
Only one slide. Huh?
Slide.
Yeah, 1 minute. Now?
No, madam.
Now can you see the slide?
Not yet, madam.
1 minute, 1 minute.
Okay, okay.
Now the slides are visible?
Okay, visible, madam, but only one slide is in front of us.
Oh, 1 minute, 1 minute.
Only the title slide is visible, madam.
Yeah, sure. I'll just. Now can you see the change?
No.
Now you can see the second slide?
No, madam.
No.
So when see about the properties of enzymes, we know that like enzyme is a protein, like we were discussing. So protein with a catalytic power, having the capability to enhance the rate of a reaction. And then this enzyme is colloidal in nature. Like when we see about, we know that when a light passes through a window or something, we see small particles suspended in the air. So those are like, that is a colloidal nature. The same way the enzyme, everything is suspended in a colloidal way. Then we have high molecular weights, that is they weigh a lot because they are made up of so many units of proteins, and proteins in turn they are made up of amino acids. They are very big, large molecules, and the substances which these enzymes act are usually called as the substrate. And on the enzyme you have got some specialized sites called as the active sites. So in the active sites only the reactions will take place. So another, when you talk about enzymes, we will know that there is something called a specific activity of enzymes. So these, when I was telling in the first few slides that we know that in the cell you have different components, right? You have the different cell components like mitochondria, you have the chloroplast, you have the Golgi complex, you have, and here in this different reactions take place, and all the compounds are present in those particular organelles. But still, nothing will go haywire, or no reaction will take place here and there. That is only because the specificity of these enzymes. One of the first thing is the stereospecificity, where they act on, there are particular enzymes which act only on the one isomer. It won't act on other. We know these isomers are compounds having the same molecular structure, but then they have the arrangement of the molecules will be different, like the L-amino acid oxidase and D-amino acid oxidase. So when it is only levorotatory, they will act. If this dextrorotatory, they will not act. So that is the stereospecificity. Then you have something called as reaction specificity. There are many substances which undergo different types of reactions using different enzymes. So when you see glutamate dehydrogenase or aspartate aminotransferase or alanine glutamate aminotransferase, all have glutamate in them, but based on the type of the reaction, the enzymatic action will takes place. The same way you have pyruvic acid, which can be acted upon by pyruvic carboxylase, where you will get carboxylation, that is you will add one CO2 molecule, one carbon to it, or you have pyruvic dehydrogenase, where it will where you have this NAD, NADP, where the dehydrogenase reactions will occur. You have pyruvic dikinase. Everything acts on the substrate pyruvate, but it is based on the type of the reaction. And the last type is a substrate specificity. So when you talk about the substrate specificity, it is again of three types. That is absolute specificity, relative specificity, and broad specificity. So only one substrate, that is, if the substrate is only, it acts only on a single substrate like urea or urease or something glucokinase, which acts on the glucose related molecules. That is the sugar molecules. And then suppose you have the relative specificity, where you have got this which act only on a particular type of like trypsin, chymotrypsin, which acts on certain proteinaceous compounds. Then you have got broad specificity like hexokinase, which acts on various number of glucose molecules. And then you know that they are amphoteric molecules. Basically, we say enzymes are amphoteric molecules because of the presence of the amino acids. In amino acid, you have an acidic group as well as a basic group. So when you have these two, they are amphoteric molecules. And then these enzymes are usually thermoliable. So thermoliable is they will act on the presence of heat. So if there is more heat is given, these enzymes polarization occurs, and if very low temperature of heat is given, crystallization occurs. That is why the enzyme urease could be extracted from JAK-B because of the ability, crystallization ability of the enzyme. So if they are given more temperature, they will denature. Then another most important aspect of the enzymes is the activation energy. See, the activation energy is that which gives the reactants the time, or it increases it to undergo the reaction in a very specific time. So when this energy is used when the reactants are converted into products, or the substrates are converted into the products, and the activation energy this increases, then the reaction will be less. If the activation energy is low, then the reaction will be more. Then when you see about the enzymes, actually previously there were different ways to express the enzymes. So they have never expressed in concentration, but they were expressed in different types of activities which they go, like King Armstrong units, Somayogi units, Reitman Frenkel units. spectrophotometric units, and they would, these are various types of units were being used. But then what happened? We all wanted a uniformity. So the IUB has given a standard unit called as the kettle. That is International Unit of Biochemistry was given it a standard called as kettle in those days. So that means that one molecule of substrate is converted to product only in a second. Whereas you have another unit called as IU unit or system international. Here what happens? Here 1 micromole is converted into product in 1 minute. So one occurs in a second and another occurs in a minute. But both of these are interconvertible. That is 1 international unit is equals to 60 microkettles. Then the turnover number. So whenever the reaction is taking place, you consider the turnover number, that is the number of substrate molecules which are converted to the enzyme, the product in a unit time. So these reactions usually, when you are sketching the enzymatic reactions, we usually put them as reversible reactions. Most of the reactions are reversible. There are few which are unreversible also, or irreversible reactions, however you call it. Then which these enzymes, they will indicate the turnover number usually indicates the catalytic power of the enzyme, the ability to convert the substrate to the product. And one of the enzymes, that is the enzyme having the highest catalytic power, is carbonic anhydrase. So these enzymes, they can be even classified based on the type of the reactions they occur. So most of the enzymes, they are endoenzymes only, but there are a few enzymes called like exo enzymes. So these the enzymes which are secreted outside the cell, they are called as exo enzymes, like some of the bacteria, yeast, mucor, and there are rhizophores. There are some insectivorous plants like sundew, Drosera or sundew, Dionia or Venus flytrap, the Nepenthes or the pitcher plant, Utricularia or the bladderwort plant. So these plants where the enzymes activity takes place when the enzymes are poured out of the plant or the substance where or the the area where it is given. Like for example, these insectivorous plants, when you take them, they are usually found in areas where you have a lot of bogs and marshes, and where the soil is not in a it's not in a very solid state. Mostly it is in a semi-solid state. So what happens under these conditions? They have very less amount of nitrogen. So the plants, because they are not able to take nitrogen, they will take the nitrogen from the insects. So that's why this Drosera, sundew, or Dionia, Venus flytrap, or Nepenthes or pitcher plants are able to get their nitrogen. But apart from that, most of the enzymes are endoenzymes. That is, they are secreted inside the cell. So wherever, even the cell also, in the certain cell organelles. Like when you are reading about the cell, it has got the outer membrane, you have got the plasma lemma, you have the plant cell especially, you have the cell wall, and inside the cell wall you have different types of cell organelles. That is the double membrane organelles as well as the single membrane. Like the double membrane like the chloroplast, mitochondria, ER, and the single membrane like whatever it is, the lysosomes, peroxisomes, glyoxysomes. So whatever organelles they are present, they will occur only inside the cell. Then based on the chemical nature, so what happened, most of the enzymes are constitutive enzymes. Only the nitrate reductase is the only enzyme which is inducible, and where the concentration depends on the substrate, whereas all the other enzymes, the concentration does not depend on the substrate, only it depends on the reactions which taking place in the enzymes. Then usually what happens, you have got some enzymes which exist in which are zymogens, where you have found they are present in inactive state, or they will have to get returned to a particular state where they will be able to be active. Usually the digestive enzymes are there, like zymogen and HCl help with the pepsinogen to convert into pepsin, which acts on the proteins and break them into smaller substances, that is peptides of proteins. And then you have got enterokinase, which converts trypsinogen into trypsin, and then you have trypsin, which converts chymotrypsinogen into chymotrypsin. So all these enzymes are there basically in the gut or in the digestive tract, where they are existing in a different form called zymogen form, the proactive form, which will again later become the active form. And then when you see about the enzymes, you should know how the reactions occur of the enzyme. So based on the reactions, they have 3 order of reactions. That is the first order reaction, second order reaction, and the zero order reaction. In the first order reaction, the reaction proceeds at the rate to only proportional to only one of the reactant, whereas in the second order it is proportional to the concentration of the 2 of the reactants. When it is in neither of the reactants, then it is called as zero order reaction. Then when you know that when you see about the enzymes, you know that different enzymes act on different substrates. So it can sometimes there will be a single substrate reaction, sometimes there will be a bi-substrate reactions. So when you see about a single substrate reaction, the reactant will immediately convert into product with the help of the enzyme. For example, you take sucrose. So sucrose, where it is acted upon sucrase, only one substrate where you are getting a glucose and fructose. So you have glucose, where hexokinase you get glucose 6-phosphate, and then you have the other examples which like there are many examples apart from this. You have like fructose 1,6-bisphosphate or 3-phosphoglyceric acid, where you get from one substrate you're getting one product.
Excuse me, ma'am.
Yeah.
Ma'am, maximize presentation. वो F5 दबा के maximize करो, madam, please.
I have maximized it, ma. Presentation is maximized, like it's on full screen in my thing.
Madam, you go for PowerPoint, madam.
Yeah, I went for power. Basically, when you have two substrate and two products, or two substrate and one product. If you want another example for two substrate and one product, you have got this synthesized reaction. Like where you have got DHAP, that is dihydroxyacetone phosphate, will condense with 3-phosphoglyceraldehyde and give fructose 1,6-bisphosphate. So you are having two substrates and one product. Okay, then again another thing is based on the chemical constitution. So where in the chemical constitutions you have got something called as simple enzymes and conjugated enzymes. These simple enzymes are those enzymes which are basically only proteins. They don't have any other additions to them, like trypsin, ligase, pepsin. They are just or urease. They are just simple enzymes. Then another enzymes, like you have got conjugated enzymes, which consist of the protein part as well as the non-protein part. So that enzyme which is a conjugated enzyme is usually designated as a holo enzyme. Means it has got an apo enzyme as well as a prosthetic group. So this apo enzyme is the protein part, and the prosthetic group can be anything, like how we saw in the bi-substrate reaction. Unmute yourself, I can hear. Prosthetic group will be a cofactor or a coenzyme. So usually when this dialysis occurs, that is the main reason where we separate the macromolecules from the micromolecules, and this is the process where you use the chemical constitution of the enzyme. So then when we are seeing about these cofactors and coenzymes, these are the additions which help in the enzymatic reactions. So these enzymes require many metal activators, like alcohol dehydrogenase requires zinc, kinases, most of the kinases which add this ATPase, they require magnesium, zinc, manganese. Aldolase, the condensation reaction, aldolase where you like for example DHAP to plus 3 PgAl, you will get 1,6-fructose 1,6-bisphosphate, where the condensation occurs, you require zinc. And catalase enzyme, which helps in the breakdown of the harmful hydrogen peroxide into harmless water, you require iron. Aquatinase, which require sulfur. So these are the metal activators which are required by the enzymes, and if the metal activators are absent, the enzymatic reactions will be delayed or they will stop. Then there are other enzymes called as metalloenzymes. See, metalloenzymes are a little different from the metal activators because these metal activators are usually external supplementations. Whereas in metalloenzymes, the metals are embedded in the enzyme itself, like Rubisco, which has got magnesium, nitrogenase, which has got iron, molybdenum, protein. So they are embedded, and if they are absent, the total enzyme collapses. Then when we are seeing about the coenzymes, the cofactors, the other thing important is the coenzyme. So usually this has a nucleotide, and then except for the iron porphyrin enzyme, usually they have vitamin B complex in them, and the substrate and the coenzyme will give the substrate A product and the coenzyme derivative, whereas the coenzyme derivative will finally give the substrate B product. So we are getting two byproducts in the bi-substrate reaction with the help of usually the coenzyme. So in the different coenzymes which we use are NADP or NAD. NADP is usually used in photosynthesis, and NAD usually in the respiration process. It has niacin, that is B5. Then coenzyme A, which has pantothenic acid. You have thiamine, B1, and you have ascorbic acid, vitamin C. And then you have FAD, where you have flavin, that is and lipoic acid. You have biotin, you have pyridoxal phosphate. So you have so many or cyanocobalamin. You have different types of coenzymes, and these coenzymes usually are helpful in the bisubstrate reaction for the enzyme to get the product. And you all the enzymatic actions, there are so many studies done on how the enzyme actions takes place. Usually the first thing was lock and key model, which was given by Emil Fischer and later explained by Paul Fitz and D.D. Woods. And then you have here what happens, they say that, when we were discussing about the enzymes, we were telling that they are macromolecules, that is they have n number of proteins, and proteins are basically amino acids, right? So in these, they say that amino acids, they will have their own orientation. The amino acid having the the carboxylic group and the basic group, all these will be having fixed sites. So when they form the active site, these amino acid groups will be fixed, and then thereby the enzyme which is having the same configuration will fit into it, and then the enzyme substrate complex is formed, and thereby the enzyme will form the product. But there are many kinds. The first thing Koshland was talking about is that he told that. Yes, Koshland was telling that that is not possible for the enzyme to always be in a very fixed state. So he has given a flexible way that is called as induced fit hypothesis, where the reorientation in the amino acids will occur and the shape is not fixed, and based on the enzyme, it will form the enzyme substrate complex, that it will be changing constantly. But so when all these things were going on, Michaelis-Menten, he has given the enzyme substrate complex theory, which involves where the enzyme, in certain enzymes it will be fixed and certain enzymes it will not be fixed, thereby justifying the enzymatic action. So when these enzymatic actions take place, they take place by different, like acid-base catalysis, substrate strain, covalent, entropy. There are different things which will occur. And the other enzymes which they saw was the allosteric enzyme. So besides the active sites, we have the allosteric sites. These sites, they are this was usually proposed by Jacob and Manor when he was talking about the one gene one enzyme theory, where he told that he was the one who given that about one gene one enzyme. So he said that apart from the active site where the enzyme active takes place, there are other things called as the allosteric sites. So where there are different modulators or effectors which bind, and they are divided into V class or Km class. So when you talk about allosteric sites, we have got different enzyme exhibiting different allosteric sites. Like you have got in hexokinase, you have the allosteric inhibitor, that is GDP. In phosphofructokinase, you have the inhibitor, that is ATP. And then you have in different parts of the enzymes, you have different inhibitors which I have shown in the slide, that these inhibitors will help the enzyme action to take place, or they will even stop. Okay, then apart from these, you have got something called as isoenzymes. See, isoenzymes are multiple forms of an enzyme which have difficult physical and chemical properties. The most important isoenzyme which slides are not changing. Slides are changing.
Excuse me, madam. Slides.
One minute.
Madam, please unmute yourself. Slides are not changing, madam. Vidya Vardhini madam garu. Madam, please unmute yourself.
Yeah, I'm just.
Yes. Yes, madam.
You can see the slide?
Yes, madam. It's visible. Okay.
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 So,
Madam, please click on slideshow. Madam,
now I clicked.
please click on
You can see?
slideshow. Slideshow. Okay, madam. Okay, madam. It's visible, madam.
Yeah. Okay, fine. So one of the most important isoenzyme which we see is lactate dehydrogenase or lactic acid dehydrogenase. So what happens here? You have got 5 forms where these are represented by 2 polypeptide chains having M or H. So when you will have 4 H's and you'll have 4 M's, or you'll have 3 or 2, or basically divided based on that we get 5 types where the M and the H will be having 2 units, having 4 units, or having 3 units and 1 unit of the other. So based on these are the which have difference based on the homologous nature or the analogous nature. Is they have simple molecular structures or they have got different structures and the same catalytic properties. When you talk about the enzymes, the first enzyme when the classification was done, it was done based on, they were divided into 2 types, that is the hydrolyzing enzymes and the desmolizing enzymes. So hydrolyzing enzymes are those enzymes which require water. So the presence of water only they will occur, like the carbohydrates, proteases, esterases and amidases. But the desmolizing enzymes were grouped into all the other enzymes, whether the removal of hydrogen atoms, whether addition of oxygen, or addition of carbon dioxide, addition of ATP, or removal of amino acids, or isomerases, epimerases and all, or hydrases were removal of. Okay. All these what happens, they please unmute yourself. Please mute yourself. Only I'll unmute. So all these which are were later on clubbed into just 2. I mean they thought that it is very difficult to put all this, so they only did it into 6 classes, and in that 6 classes they have divided each class into 4 to 13 subclasses, and the subclasses were divided into subclasses, where the first digit was the major class, the second digit was a subclass, the third digit was a subclass, and the fourth digit was the serial number of the enzyme. So they have given even serial numbers of the enzyme. So these days when we are writing anything about enzymes, we should be very specific and give the serial number. So like alcohol dehydrogenase, which belong to oxidases, so it is in the first thing. So it is 111, where it is having oxidases, where it is only the oxidation, that is a redox reaction occur, and that was even giving rise, when we are talking about these things, they are even giving rise to the naming. So the nomenclature of the enzyme was also considered very important. So where usually most of the enzymes, most of the enzyme, the name was ended with AST, that is ACE. So each enzyme, what they did, they consisted of 2 words, that is the name of the substrate and the name of the reaction. For example, hexokinase. So hexokinase is the first enzyme used in glycolysis, where glucose is phosphorylated to glucose 6-phosphate. So where ATP is transferred. So hexokinase was renamed as glucose 6-phosphate ATP transferase, because it was transferring ATP and it was forming glucose 6-phosphate from glucose. And then if there was, other than this ATP, like ATP transferase or oxidation or something else, 2 reactions occur instead of 1 reaction. 2 reactions are occurring. Then whatever the extra thing was kept in brackets or the parentheses. Like pyruvate dehydrogenase was renamed as pyruvate NAD oxidoreductase. So here the oxidoreduction is taking place. That is NAD. NAD is also oxidoreduction reaction is taking place. Apart from that, 1 carbon dioxide also is being released. So because the compound is losing 1 carbon dioxide plus an oxidation reduction reaction, the addition reaction whichever is taking place is kept in the bracket. Then the major classes which were there are oxidoreductases, transferases, hydrolyses. In oxidoreductases, these are the first 3 classes. The oxidation of which occurs in CH-OH group, CH-CH group, or C-double bond O group, like alcohol, NAD oxidoreductase, cytochrome C oxidase. So wherever the oxidation or the oxidoreduct reaction are occurring, they clubbed it. That is, in the when you see the previous thing, so all the dehydrogenases and the oxidases were clubbed together, and peroxidases were clubbed together into the major class oxidoreductase. Then you have got the transferases. In transferases, the transfer of the aldehyde, the keto, the carbon, amino, phosphate. So all the transferases, that is amino transferases, ATP transferases, all were clubbed and they were put, that is those 14 groups were just put again in the 6 major classes. So they like glutamate, glyoxylate, glutamate, amino transferase, phosphofructokinase, transketolase, all these enzymes were clubbed into transferases. So then hydrolases are whichever requires water. That is the first 4 hydrolyzing enzymes were clubbed and they were put into hydrolases like sucrase, maltase, protease, fructase, which acts on fructose diphosphatase, glycerol ester hydrolase. So all these enzymes were clubbed and put into the hydrolases. Then again the next major class is lyases. So what these lyases do is they break down the CO, C and CC bonds, and then they, like for example, 3p carboxylate, pyruvate decarboxylase. So lyases, whereas ligases will join. They're acting in the opposite way. Whereas isomerases are just those group which will help in the intermolecular reactions. We're like triose phosphate isomerase, phosphofructose isomerase, phosphohexo isomerase. So when you talk about the other property of enzyme is the competitive and the non-competitive and the feedback inhibition. Okay, so when you see about the competitive inhibition, you have, all of you must have heard about the or read about the enzyme nitrogenase, where oxygen is a competitive inhibitor. So if there is more oxygen present, then nitrogenase activity will reduce. So it is competitive inhibition. And here in competitive inhibition, so in competitive inhibition, what happens? The enzyme activity will stop because they will be competing with one another, and then the molecules also will stop the reaction if there is excess. So for example, when you see hexokinase, if there is more of glucose 6-phosphate, then hexokinase activity will stop. That is called as